study the structure of DNA.
DNA stands for deoxyribonucleic acid, and its main function is to carry hereditary information in many organisms. You must heard about the double helix model of the DNA that demonstrates that DNA is basically a polymer.
So, DNA is a long polymeric chain made up of nucleotides twisted around each other in the form of a double helix, and the length of the DNA is as well defined as the number of nucleotides present in it.
A nucleotide consists of three important components: a nitrogenous base, a pentose sugar, and a phosphate group. The nitrogenous base is linked to the pentose sugar through beta N glycosidic linkage to form a nucleoside through first carbon of sugar and nitrogenous base. There are two types of bases: purines and pyrimidines. Purine is a 9-membered ring made up of two rings, one pyrimidine and one imidazole ring. Purine include adenine and guanine, while pyrimidine is a 6-membered ring and it includes cytosine and thymine in case of DNA, and in case of RNA, it includes cytosine and uracil. According to Jager's rule, concentration of purine is equals to total concentration of pyrimidine. In case of purine, N9 is linked to the pentose sugar, and in case of pyrimidine, N1 is linked to the pentose sugar. The beta N glycosidic bond is formed by removal of elements of water, a hydroxide from sugar and a hydrogen from the base. In case of pentose sugar, 4 carbon atoms are combined with one oxygen to form a ring, and the 5th carbon atom forms CH2 group outside the ring. The only difference between the two pentose sugar in RNA and DNA is that there is one less oxygen atom in deoxyribose at 2'. Do you know the backbone of a polynucleotide chain is formed due to a sugar and phosphate? Phosphate group is linked to the 5' modes of the nucleoside, and a phosphoester linkage is formed. The strong negative charges on the nucleic acid is due to the presence of phosphate group. Bond between two different phosphates form phosphoanhydride bond, and these bonds are thermodynamically more active than the phosphoester linkage. Two nucleotides are linked through 3', 5' phosphodiester linkage to form a dinucleotide. Now, let's talk about some features of the double helix structure. The double helix structure is made up of two polynucleotide chains which have anti-parallel polarity. That is, one is from 5' to 3', and another one is 3' to 5' in direction. The bases in the DNA are paired through hydrogen bonds between A and T and G and C, where G and C consist of 3 hydrogen bonds, while A and T consist of only 2 hydrogen bonds. That's why if GC content is more, then the DNA denaturation temperature is also high. You must be noticed here always a purine comes opposite to a pyrimidine. Actually, this generates uniform distance between the two strands. The plane of one base pair stacks over the other in a double helix. This, in addition to hydrogen bonds, confers stability of the helical structure. Hydrogen bond is important for specificity of base pairing as it provides the thermodynamic stability. The two chains are coiled in a right-handed fashion. The pitch of the helix is 3.4 nanometer and there are roughly 10 base pairs in turn. The twisting of the double helix creates several more interesting features in DNA. Firstly, the groups are formed by the twisting are not uniformly spaced. One is called the major group, which is 22 angstrom wide, while the other one is minor group, which is 12 angstrom wide. These groups often provide the binding sites. The diameter of the helix is about 20 angstrom.
So, this was all about the basic structure of DNA. I hope you liked the video.
If you like the video, please like and subscribe my channel.